Did you know that fungal infections kill more people than TB or malaria, which is why it's so important for more research to study about these fungal infections and how we can make a difference? So my name is Hanna Zafar. I'm a second year PhD student at the Institute of Microbiology and Infection at the University of Birmingham. This institute is one of the largest worldwide of microbiologists all studying to try and find ways on understanding infections. Specifically, I work on fungal infections, and in particular, I'm studying Cryptococcus neoformans. It's a basidiomycete yeast, which is a fungus, and it mainly infects people who have HIV infections, specifically people in sub-Saharan Africa. There are 220,000 cases worldwide, and 181,000 people can die from this disease, which is why it's so important to understand the causes of this fungal infection and how we can make a stop to it. 60% of people can actually become drug resistant to this particular fungus, which is why I'm studying the drug resistance aspect of this fungus. So right now, I am currently running I'm planning to run a gel. So I'm going to be doing some gel electrophoresis. And it's a molecular biology tool that is used to see DNA in our samples and how big or small they are, just to identify the DNA. So right now, I have my samples here. I had to pre-make the gel because it takes some time to solidify. So right now, this is one of the gels. It's a 0.8% agarose gel. We use these combs to help make the wells inside the gel, so to add our samples into the gel. So right now, I'm going to load my sample into my gel. The way to do that is, the first thing you do is you add some loading dye into our sample. This just helps bind to the sample and make sure that it runs through the gel and helps us to visually see it as well. So I'm just going to get that. So right here, I'm just going to try and get some. Sometimes it's necessary to look at other bottles that have the stuff. So yeah, just going to get some loading dye. Add a little bit to my sample. So just a little bit. And then I'm just going to mix it using the same technique, so just mixing it up and down. So again, I'm just going to put it up and down just to mix it thoroughly. Take it up, and then I'm going to add it into the well. So here you can see that there's a red strip underneath the gel. That just helps me visually see where my bands are, so when placing them I don't have to poke around to have a look. So I'm just going to insert it into this well right here. And that's me adding the sample in. So the reason I decided to do my PhD in medical mycology is because I realized that there is not enough awareness about fungal diseases. It's just as important as other infectious diseases such as bacteria and viruses, and I do believe that more research is needed to understand these infections so that we can help discover the drugs and see how to help people who suffer from these infections. I hope this was interesting for you, and if you have any questions, please leave them in the comments below, and thank you for watching.